Welcome, everyone. I know we have a lot of faces from UCI that are here tonight, but trust me when I say I'm not just speaking to you, but I'm speaking to our YouTube channel audience because we record for these, as many of you know. I'm really pleased that so many people came. It's been, I think everyone's heard me say this, it's been tough coming back from COVID and building up an audience again, so it's really nice to have people coming out and participating. I'm Eileen Anderson. I'm director of the UCI Stem Cell Research Center. This is our evening community lecture series, which we host once a month, something like 6 or 7 or 8 months of the year. A couple of quick things for the center in terms of just exciting news. I always try and tell something that's good. Despite the fact that California Institute of Regenerative Medicine is a little understaffed right now and has a pause for their clinical trials program, their CLIN2 clinical trial program, we actually have super exciting news, and that's that 2 of our investigators have new investigational new drug applications that have been approved to go forward. So we're just waiting for that next stage of CIRM funding to come. So that's Leslie Thompson with an IND for Huntington's disease, and also Daniela Boda with an IND for glioblastoma, brain cancer. And that's really exciting for us because UCI has been building this basic science, many of you have heard me talk about it, to translational science to clinical pipeline. So this is really our first 2 all the way through investigator-initiated programs that are going to clinical trial. We have almost 60 clinical trials that are on track in our CIRM-funded Alpha Clinic, but many of those are from companies, from other sites. These are the first 2 that are going to roll all the way through out of UCI. So we're super proud of them, and we want to congratulate them. With that, as my one exciting piece of news tonight, I just want to welcome our speakers. And in my worst example of I have been drinking through a fire hose this year and I'm completely unprepared, I would like to invite our speakers up front, and I'm going to put on my first director Peter Donovan hat and torment them with random questions, because that's what he would do when he was unprepared, but he was so good at it. Really impressive. So, Katie Thompson-Peer, so pleased to have you here tonight. Why are you a scientist?
Why am I a scientist? Well, first of all, hello. My name is Katie Thompson-Peer. I am delighted to be here. I'm a scientist, I think, because I like troubleshooting and I like solving problems. I like thinking about data and what the possible interpretations could mean. And I like working with other people to try and figure out what do we do next about this particular problem. I'm very curiosity driven. So I want to know why something works and how something works. And I want to work with the people who are in my lab and in my community to pick apart those problems. So I think I'm a scientist because I like the process of doing science.
That's awesome. And what department are you in?
Oh, I am in the Department of Developmental and Cell Biology here at UCI, which is over in the School of Biological Sciences. I am associated here with the Stem Cell Center, as well as a couple of other centers here on campus. I'm an assistant professor, and I've been here for about 5 years.
Are you biting your nails now? No. We knew, like, from the first time I met Katie, we knew she was going to be a success here, so no problem. Sydney Prang, did I say your last name right?
Yes.
Why are you becoming a scientist?
Hi, I'm Sydney. I'm a 4th year PhD student in Katie's lab. I'm becoming a scientist because I think science is really fun and exciting. And I feel like being the first person to see something or discover something is like really gratifying and just exciting. And I'm pretty much doing it because it's fun. So I think it's like the coolest job that you could have. So yeah.
Yeah, you will be successful because you pretty much have to think this is the coolest job you'll ever have.
Yes.
All right. So with that, just as a quick icebreaker, please feel free. Can I invite people to ask you questions as you're doing your presentations? I think those are 2 really kind of useful answers to give. And the reason is that I just talked about the culmination of what happens when you do research, right? And that's that we move all the way through discovering something new that we can get excited about, to troubleshooting it, to actually trying to apply it. And that's what happens when we move into clinical trials. We're in the application stage. There's actually one step more than that, because clinical trial is a human experiment, right? reducing that to practice is the ultimate culmination, right? When we can give someone a cholesterol drug and, half the population is on cholesterol drugs, we've reduced it to practice successfully. But all these different pieces leading up to that are important, and it takes people that are just wanting to come to work every day because they're enthusiastic about it to get it done. So with that, can I invite you guys to start your presentations?
Okay. Thank you for that introduction. Thank you all for coming. I'm so excited to have you here. I'm going to tell you about the kind of the big picture of what my lab studies and the questions that we're interested in. And then Sydney is going to tell you about her particular project and how that kind of fits in with the big picture. So our lab is interested in the nervous system, which is composed of the brain and the spinal cord, your central nervous system, and as well as the peripheral nervous system that innervates all of your tissues. And we're interested both in how the nervous system develops, but really in how the nervous system responds to injury and how it recovers after injury. So if we look at the cells of the nervous system, there are a variety of different cell types. Nerve cells or neurons are the primary electrically active cell type in the nervous system. They're not the only one. There are glial cells of various kinds and other cells that serve various essential functions for the nervous system, but we're primarily interested in the nerve cells that transfer information from one part of the nervous system to another. And so if you look at an individual nerve cell, here I'm showing you a picture of one of the nerve cells that we study. There's, you got the cursor. The nerve cells have a couple of different kinds of projections, couple of different parts of the cell that stretch out into the world that are essential for how they function as part of a circuit. Because all of the nerve cells are going to come together to be how information is processed, how you receive information from the world, process that into your brain, and communicate that into actions and behaviors that you engage in. So if we're looking at a single cell, there's the cell body, the cell soma, which has all of the DNA and performs many of the functions of the cell. There are these highly branched structures that you can see. There's the cell body in the middle. There's these highly branched structures all around the nerve cell, all around the neuron. These structures are called dendrites, and those function as the receptive antenna for the nerve cell. That's how nerve cells receive information. So if you're talking about neurons that are out in the peripheral nervous system, that's how you sense touch or temperature. If you're talking about nerve cells that are in the brain, that's how they receive information from other nerve cells in the brain, is along these highly branched structures called dendrites. That's information in. That information in the form of electrical activity is processed by the neuron in the cell body, and then the neuron decides to send information to other parts. So if there's a nerve cell that needs to activate a muscle to cause a muscle to contract, there will be a signal that is sent out of the cell along this long cable-like process that's called an axon. And axons can be very long. There are axons that stretch from your spinal cord all the way down to your toe that are important for how nerve cells send information out, right? Okay, so we have information coming into the nerve cell through these dendrite branches, this whole complicated arbor, and we have information going out of the nerve cell along this axon. And so what I became really interested in were how do these two different parts of the nerve cell respond to injury in similar or different ways? How do neurons respond to injury to their dendrites versus how do neurons respond to injury to their axons? And how do they recover after those injury processes? And unfortunately, neurons can be injured in people in a variety of different conditions. Spinal cord injury is one that we think about commonly. It's a failure of axons to regrow along the spinal cord after that's damaged. You can also have damage to the brain, thinking about stroke or traumatic brain injury that are going to cause massive cell death, but then also cause injury to the dendrites of the cells in the brain. There can also be injury to neurons, not just in the central nervous system, but in the peripheral nervous system. So if you have peripheral neuropathy, that is the dying back of neurons that innervate your tissues. That happens a lot to people who have diabetes. This happens to a lot to people who undergo chemotherapy. They have a dying back of the neurons that innervate their tissue. And so then they lose feeling or lose other sensory abilities for their peripheral nervous system. And so unfortunately, neurons are going to get injured. And so we wanted to know, how can they recover from this? And I was initially going to start off this question doing kind of a compare and contrast, looking at the literature to see what do we know about how neurons respond to dendrite injury versus how do neurons respond to axon injury. And that's when I stumbled across kind of a black box within the scientific literature, because it turned out at the time, this is a snapshot of the scientific repository of papers that have been uploaded to PubMed, which is the National Library of Medicine. That's where a lot of academic research gets deposited. And at the time, this was about 10 years ago, there were 1,200 papers on axon regeneration. So how do neurons regenerate axons if they are injured? But there were only 3 papers on dendrite regeneration. And it turns out that this one is a commentary on another one of the papers. So there were really only 2 papers in our scientific literature about dendrite regeneration. And so you can't really do a good compare and contrast if you don't know anything about the one side of the equation. And I just told you that dendrites are essential for neuron function. They're how they receive information from the world around. And so I was really confused and perplexed about this for a long time. I went to a bunch of conferences to try and see if there was a scientific literature about it that I couldn't find. Maybe I wasn't using the right keywords. Maybe people had studied this and described it in different ways. And I got a lot of responses of people saying, I don't know about dendrite regeneration, but here, let me tell you all about axon regeneration, right? Which is a very interesting and viable part of science to study axon regeneration, but there was this black box about how do neurons regenerate dendrites. And it seemed like people just really hadn't designed the appropriate experiments to study those questions, I think for a number of reasons. So I went back, way back in the literature, all the way back to Santiago Ramon y Cajal, who was a scientist who studied in the late 1800s, early 1900s. He's one of the fathers of modern neuroscience. He did all of these beautiful stainings and drawings of nerve cells. And it turns out if you go all the way back to his literature, he had looked at dendrite regeneration in addition to looking at axon regeneration. And he's got these beautiful drawings of nerve cells. There's the cell body in the middle, and he's got some dendrites going up, and there's a scar tissue up above. And he's trying to examine, is there any evidence of dendrite regeneration in this particular cat that he had been looking at? And he didn't find any evidence for it. And so he made all of these conclusions about dendrite regeneration and then also axon regeneration. And the field kind of took his work on axon regeneration and ran with it. And people didn't really follow up on his work looking at dendrite regeneration, I think for technical reasons, because it's very hard to do these very precise scalpel-based injuries that are injuring these dendrite branches that are right close to the cell body. And so we decided we wanted to take a look at this question about how well neurons could regenerate dendrites, but take a much more modern approach, where we're going to use microscopy and lasers to specifically cut off dendrite branches and ask really basic fundamental questions about whether or not neurons could regenerate dendrites or not. Like, was it even possible for neurons to regenerate dendrites or not? And furthermore, if they can regenerate dendrites, how well do they do? Are neurons capable of perfectly regrowing this complex, highly branched arbor, or when they are tasked with regrowing after injury, do they somehow fall short? Are they capable of regrowing, but it's in some way disorganized? And so that's where we started. And we started off, as I said, looking at these neurons using a 2-photon laser to selectively and specifically cut off all of the dendrite branches, right? So the cell body is in the middle and the axon's down, and we use the laser to sever off all of the branches. And if you come back and look at the same neuron 24 hours later, this is what it looks like on the left. There's the cell body in the middle, and the axon is going down, and you can see that it's starting to regrow a few little branches. And we come back a couple of days later after that, and this is what that same neuron looks like. There's the cell body in the middle and the axon going down, and you can see that dendrite regeneration is really robust. These neurons are regrowing a ton of branches, and they're regrowing, in fact, maybe similar to the right number of branches. If I show you side by side here on the left, this is our uninjured neuron, and here on the right, this is a neuron that is 3 days after we cut off all of the dendrite branches and allowed them entirely to regrow. They regrow a ton of these projections. When we go through and count all of the number of dendrite branches, they regrow the right number of dendrite branches. So if they're supposed to have about 350, they regrow about 350. If we look in other cell types that are supposed to have 35, they regrow 35. Yeah.
What animal is the cell in? I mean, it's a mammal, right?
No, we're not looking in mammals. So we use Drosophila. We use fruit flies as a model system to study this. The reason that we use fruit flies is because we can follow individual neurons as they are responding to injury over time. So we can look at a single cell and damage it, and then come back the next day and look at that exact cell and look at all of the branches, and then come back a couple of days later and look at the exact branches. And we can do that in the intact normal tissue, right? So I think one of the complexities that these cells are looking at when they're regrowing is, how do these dendrites sample space? How do they sense the tissue that they are regrowing into in order to regrow a new dendrite arbor? And so that's why we wanted to do it in a normal intact organism. So we're not damaging any of the rest of the tissue. Okay? So they regrow a lot of dendrite branches, but there are a number of ways in which dendrite regeneration, we found, fell short of what a perfectly normal uninjured neuron would look like. The territory is a whole lot smaller. If you measure the size of how big these branches are, it's about half. The dendrite branches cross over one another a whole lot. They're not supposed to do that. So a normal uninjured neuron, if we look at the places where the branches cross over one another, it does it very rarely. But if we looking at these neurons that are regrown after injury, they cross over each other a ton. So they ignore these cues in space that determine where they are supposed to grow. And so we wanted to understand kind of two sides of what is happening here. How is dendrite regeneration as robust as it is? Because every neuron that we've studied, if we damage dendrites, it starts regrowing. It's very reliable across lots of different kinds of cell types. But then also, why is it as disorganized as it is? What is it about the cell? What is it about the tissue that it means that it can't perfectly regrow what it had lost? It can't perfectly recreate what an uninjured neuron looked like. And so that's the kind of the stories that my lab is interested in, is both how do neurons sense this particular type of injury in order to mount a robust regenerative response, but then also why is it so disorganized? So one of the stories that my lab has been looking at is, how do neurons sense this type of injury? We've got a story coming out soon that we think suggests that the neurons use an influx of calcium, the influx of the ion calcium, to detect the fact that they've been injured. I'm going to show you two neurons here that are expressing a construct that means that they glow green when there's high levels of calcium. And we've got a neuron on the left that we're going to cut an axon, and a neuron on the right that we're going to cut a dendrite. And I'm just going to show you the movie. Initially, this neuron is pretty dim, but we're going to cut an axon down there, and you can see the whole thing glows right up. There's a whole lot of influx of calcium right after axon injury, and we see something that is in some ways similar to when the neurons experience dendrite injury. So over here, we're going to injure a dendrite, and you can also see that the whole cell lights up, that we get this influx of calcium that we think could be part of how these neurons are sensing that they've been injured. So that's one story that we've got going on. We've got more work that looks at, how do these neurons, when they are growing out, sample space? How do they interact with the other cells and tissues in that environment when they are regrowing? So here I'm going to show you a time-lapse video of a neuron where we are going to take one picture every minute. This is a zoomed out view. There's the cell body in the middle and the axon going down and all these dendrites. And I'm going to show you also a zoomed in view of this on the right. And again, there's going to be one image every minute. And what you can see is that these dendrite branches are highly dynamic. They're growing and shrinking. They are running into each other, they're retracting fully, they're engaging in all of these very active behaviors that determine what the shape and what the function is of these nerve cells. So we want to know, what is it that these nerve cells are sensing? It turns out that the nerve cells, when they're regrowing after injury, are a whole lot more active than the uninjured neurons. So how is it that we got neurons that are much more dynamic after injury? A third story has to do with how do the cells move the cellular components around in the neurons that are responding to injury. So here we have a green fluorophore that is showing the outside of the dendrite branch, and we have a red fluorophore that is going to show you an organelle called a mitochondria that moves around in these dendrite branches. And you can see that these mitochondria crawl around in the dendrite branches and make choices about where to go left or right, how much to move. And we see all of these changes in the mitochondria after dendrite injury that might be responsible for how the neurons are able to provide enough energy to support the kind of monumental cellular task of regrowing all of these dendrite branches. So altogether, my lab is interested in these questions of how do neurons sense dendrite injury? How do neurons mount a regenerative response? And then how can we leverage the fact that it is as robust as it is in order to teach us more about cellular biology of these nerve cells and their recovery after injury. And with that, I'm going to introduce my graduate student, Sydney, who's going to tell you about her project. Yes.
Quick question. In the regrowth of the dendrites, what's the average length? Is it in millimeters or microns?
It's in micrometers. Yeah. So the length never recovers. So even though these neurons regrow the right number of dendrite branches, it's a whole lot shorter, right? So if you look at the total length that the arbor is able to regrow, it depends on the cell type, but it's usually somewhere around 60% of what a normal neuron would look like. That is reflected in the fact that the function of the neuron only comes back about 60%. Okay, yeah?
If you whack off the dendrites in a young animal versus an older animal, they restore to 100% instead of 60%?
They can never restore to 100%, but if you get older and older, they regenerate less well. So if we look...
I don't like that answer.
So if we look in the youngest animals, they regrow somewhat more effectively, but it's never perfect. And the defects in terms of like crossing over one another happen at any point. We've got some experiments where we're looking in older animals that are getting to be really, really old to see if there's ways that we can improve the regeneration in much older animals. That story is not yet ready for prime time, but I am interested in trying to figure out how can we manipulate like genetic pathways that control aging in order to enhance regeneration in those older animals, because there's absolutely a decline in regenerative potential. Okay.
I'm a little bit shorter than Katie is, so I have to make it a little bit shorter. Hi, once again, I'm Sydney. I am a fourth-year PhD candidate in Katie's lab, and I'm also a CERM training grant fellow. And my presentation is going to be a little bit more of a specific story about neurodegeneration. Because, we see this robust regrowth in these of dendrite arbors following injury, but we kind of wanted to know what would happen if an unhealthy neuron was injured? Can we get the same response? And can we make that better? So to answer this question, we wanted to look at neurodegenerative disease and model that in our neurons. So across different neurodegenerative diseases, you get a hallmark across all of them is dendrite defects and dendrite loss and dendrite degeneration that happens relatively early on in the disease progression. So here in a mouse model, these neurons, this is what they normally look like in the striatum. But if you have a Huntington's disease mouse model, you lose a lot of the these things called spines on the dendrites, which are those little tiny spikes along the dendrite branches, and you lose a lot of those in a Huntington's disease model. Similarly, if we look at a mouse cerebellum, this is what the neurons and their dendrites normally look like. But if you look at a model of spinocerebellar ataxia type 1, you get this sort of altered dendrite morphology, loss of dendrites, and dendrites not being where they're supposed to be, as well as just general loss of the complexity of those dendrites. Similarly, we see the same thing in postmortem patient tissue from humans. So this is an aged neuron in a human brain. And that same sort of neuron in a person with Parkinson's disease looks like this, where you really have that loss of dendrite branch complexity, loss of those spines, and loss of dendrite branches in general. Similarly, if we look at a neuron in the hippocampus of an aged person, they have this nice dendrite arbor and all these spines. But if you look at an Alzheimer's disease patient of someone around the same age, you also have this loss of dendrite branches and this loss of these dendritic spines. So across all these different neurodegenerative diseases in humans, in animal models of these diseases, we have this loss of dendrite complexity and loss of dendrites that is a result of the disease. And notably, this happens relatively early in the disease progression. So before there's any mass neuronal death, which is generally associated with later stages of neurodegenerative disease. So we were thinking, okay, if these neurons are, fated to degenerate and eventually die, sadly, maybe we could kind of target this early stage during that dendrite degeneration process and see if we could either reverse or delay that by harnessing the power of regeneration. And we do that by, looking at the neurons in our system. And one of the sort of easier diseases to model in our system is polyglutamine disease, which is a type of neurodegenerative disease that is caused by expanded CAG repeats in the genome. These include diseases like Huntington's disease, which a lot of folks here study. And in a healthy person who does not have the disease, you'll have a gene that just has this standard number of repeats, and that creates the correct protein that it's supposed to be. You get a healthy neuron that functions properly and a healthy nervous system. However, if you have someone who has an abnormal number of these CAG repeats, CAG being glutamine, which is an amino acid, you end up with an abnormal protein because you have an abnormal number of those glutamine amino acids, and then that leads to issues with neuron function and neuron degeneration, and then eventually an unhealthy nervous system and progressive death of neurons. So since our lab really is kind of interested in neuron cell biology, we wanted to try to see if we could kind of stop this process at the neuron level. And when we model these sorts of diseases in the neurons we study, in young animals, they look relatively healthy. On the left, you have the control neurons. They have this nice, beautiful, robust dendrite arbor. And then in our different polyglutamine disease model neurons, they don't look that different from a normal healthy neuron. However, as the animals get older, the healthy neurons continue to grow that dendrite arbor, and it gets really robust and really large and really complex. But all of these different disease model neurons, they start to lose that dendrite complexity and start to lose dendrite branches. And they really aren't filling the space properly or any of those things. So this provides us with a really nice model to study neurodegeneration and how we can look at regeneration as well. And here's just a sort of overall quantification of what that looks like. So our wild type neurons, the healthy neurons, they grow over time. But then with these neurodegenerative disease model neurons, they grow to a point and then they start to decline in terms of dendrite branch number. So these neurons that are fated to degenerate and then eventually die, we wanted to know what would happen if we induced an injury at that time point between when the neurons are relatively looking similar to a healthy neuron and between when they start to degenerate. So what would happen if we injured these neurons? Could we induce regeneration that would sort of combat the degeneration that we see? And surprisingly, we did have a very exciting result. Here, once again, is just an uninjured neuron on the left, and we quantify this by normalizing it. So we just basically pretend that this branch isn't there so that we can effectively compare it to a neuron where we've removed an entire branch. And hopefully what you can appreciate is this neuron where here I remove the dendrite branch. This neuron looks quite a bit more branched than the neuron on the left. So this is the neuron where we actually induce the injury. And you can see that there's a bunch of these little tiny dendrite branches that are sprouting up across these big long branches, whereas in our uninjured neuron, you don't get a lot of that. It's not complex at all. And this phenotype that we saw was recapitulated across all of these different disease model conditions. So in our spinocerebellar ataxia type 1 model, we see that the injured neurons have more dendrite branches in the model for SCA2, SCA3, and our Huntington's disease model. So across all of these different neurodegenerative polyglutamine diseases, we can induce this sort of neuroprotective effect where these neurons have more dendrite branches than an uninjured neuron at that same time. And we kind of wanted to further look at what was actually happening in that time point after injury. So if we look at our uninjured neurons, they're overall losing branches. You have this negative trend where the majority of neurons are losing branches during this time, but our injured neuron is basically stabilized. So overall, the general growth trend is that there is no decline and no increase, which effectively is rescuing this degeneration. And instead of those neurons, being faded to really start to lose those or continue to lose those dendrites, the arbor stabilizes and protects what is still there. And once again, looking at these neurons, right, that are faded to degenerate and die, what we found is that if you induce this injury to a single dendrite branch, you get this protection of those remaining dendrite branches instead of that progressive strong degeneration that we normally see. As Katie mentioned, dendrite regeneration is a rather small field, so we kind of wanted to also look at what would happen if we injured axons. So whether this was just a result of general regeneration being turned on in these neurons, or whether it was specific to dendrite regeneration as a process. So we looked at what would happen if we injured an axon in these neurons. And not really surprisingly, but good for us because we really care about dendrite regeneration, we did not see the same effect. So here's our uninjured control neuron. It really does not look very healthy. It has not a very complex dendrite arbor. It's really not filling the space it needs to. And when you look at a neuron where we introduce an axon injury, so that dotted line is where we remove the axon, those neurons really don't look all that different from one another. And when we quantify that out, it turns out that that's true. The number of dendrite branches is not different between the uninjured and the neurons where we did the axon injury. And once again, when we look at what's actually happening after that injury, the uninjured neurons are degenerating and the neurons where we did the axon injury are also degenerating. So it doesn't provide that protection in the same way that the dendrite injury did. So overall, these neurons that are fated to degenerate and die, when we do an axon injury, we don't get any protection of those neurons. But when we do a dendrite injury, we are able to protect some of those remaining dendrite branches from degeneration. And really what I've been focusing on for like the last year or so is really what's actually happening to these neurons. We don't know a lot about dendrite regeneration and the processes that are at play. So this, provides us a really good way to actually kind of dig into and investigate that, and also to see what's allowing for this protection to occur. And so one thing we wanted to look at first, and I'm now realizing my slide is missing the text, which is why it's blank, but we wanted to know what was actually happening to these neurons. And one thing we wanted to look at first was this protein called actin, which is really important for neurons to maintain their structure, and it's also important for neurons to make new dendrite branches. So we wanted to see what was happening to actin in these neurons. If you look at an uninjured neuron, basically we're just marking the relative levels of actin in that kind of purpley color. So as the color goes from more purple to more yellow, that means there's more of that protein there. So in a normal uninjured neuron of our neurodegenerative model neurons, there's relatively low levels of actin. Actin is perturbed in these neurons normally when they are degenerating. But when we induce the single branch injury, you get this really bright increase in actin after that injury. And this is happening just 24 hours after injury. So even before the time point where we were seeing like that protection. So this seems to suggest that it's part of the mechanism of that protection. It's increasing the neuron in response to injury increases actin, and that allows for that protection to occur. Similarly, when we looked at the same thing with axon injury, we don't see that increase at all. So this really suggests that this is a specific process that's being caused by dendrite injury, and during the dendrite regeneration process, you get this increase in actin, and that is what's allowing us to have this protection. And this is just quantification of that, but overall, for all of the different models, we saw the same thing, where in the single branch injured in like the green, you get this increase in actin in both the cell body and in the dendrite branches, but for the axon injured neurons, you don't get that. Sometimes you get a decrease in F-actin, sometimes there's no difference. So overall, what we're thinking is that with these neurons that are fated to degenerate and die, we can't really save them with an axon injury, but the promise of dendrite injury and dendrite regeneration is that we are able to protect those neurons by turning on regeneration via injury. And that process is sort of supported by this increase in F-actin throughout the neuron. So ideally, you don't want to have to injure the neurons of someone who is suffering from a neurodegenerative condition or who is experiencing neurodegeneration. So ideally, what we would like to do is, instead of have these neurons degenerate, we can really piece apart what's actually happening in this process so that we can induce that regeneration and that protection without having to, shoot a laser at anyone's head. So yeah. And these are all the people that helped me with this project. My undergrads, Isha, Anjali, and Lee, have been really instrumental in quantification of this, as well as a former undergrad, Daria, who also helped a lot with this project. I'm also very thankful to CERM for funding me for 3 years on their training grant. I've learned a lot, and I've got a lot of experience being able to talk to like clinicians who work with folks who are have these neurodegenerative conditions, and getting to really see like the clinical side of it, because obviously, we're working in flies, so I don't really get to see that very often. So I'm grateful to CERM for that, as well as the Rose Hills Foundation, which also funded me, and Katie's R00 from the National Institute of Neurological Disorders and Stroke. Yes.
Let me bring you a microphone. For the neuroprotection studies, did you look longer than 3 days? Is the protection sustained?
That is a good question. Unfortunately, these are a bit of a terminal experiment, because we are doing all of these experiments in Drosophila, so fruit fly larvae. And after 7 days, they turn into a pupa so that they can become an adult fly. So I haven't been able to look past that time point, because they degenerate quite late in larval life. So that's basically the last point I can look at. But definitely something I want to do in the future is potentially look at this sort of response in adult neurons. Unfortunately, these neurons die during pupation, so we can't really look at them past this time. But I think looking in adults, we'd be able to answer that question about whether it's sustained or not.
Great question. But this would be a good point, or a good point in the discussion, at which I think I'm going to point a question at you, Katie. And maybe just have you, chair's prerogative, say a few words about the great question we had earlier on the model system that you're using. So you gave a great answer about the ability to see stuff by using flies that we can't see in a mammalian system, right? But maybe you could say a few more words about model systems and how much they help us to understand and how much biology is conserved, right, across them.
Right. Yeah. So, and I think that, thank you for that question. Drosophila has been a kind of major system in which we have used, scientists have used to understand processes of basic biology. So when we're looking at the function of a nerve cell, the basic cell biology of a nerve cell in Drosophila or in other model systems is highly conserved to the basic cell biology of how nerve cells function in mice and rats and humans and people. So I think that the strength of the model system is that we can kind of tease apart a lot of the basic cell biology in a way that where it's really experimentally accessible and look for novel ideas in terms of how these cells function, how they respond to injury with a very precise injury with a lot of experimental ability to kind of tease apart that cell biology. I think everybody has their kind of favorite examples of how model systems in general have been used to understand disease and that's relevant to humans. My personal favorite is the fact that when there was a research scientist who was studying sea urchin development, and when he was studying how sea urchins develop and how their cells divide, he was able to identify these proteins that change as the sea urchins develop and as they divide. And he discovered this set of proteins that are called cyclins that are absolutely essential for the cell cycle. And it turns out that cyclins and the disruption of the cell cycle is what goes wrong in cancer, where you have exuberant cellular division. And so I always think about Tim Hunt, who was studying sea urchin development, understanding these kind of basic principles of cell biology that really then went on to open up our understanding of what goes wrong when cells aberrantly divide and develop. And so I think that the promise of model systems research is experimental tractability that can lead us down these unexpected pathways, but really reveal insight into how these cells respond to injury.
Perfect. I saw her first. Sorry.
Hi, great talk. I'm wondering if you know, if you injure a dendrite, does it prime the neuron to be more likely to regenerate its axon?
Do you want me to take that? Sure. Okay. So far, we don't think that it does. I spent a while looking for evidence of a conditioning injury kind of priming event, looking between if it was injuring two dendrites, injuring the one axon versus another, versus an axon versus a dendrite. I haven't found specific evidence in this case that there is crosstalk between these two different kinds of regeneration, right? It doesn't mean that it's not there. It just means that so far, all of the experiments that we've looked at suggest that the neuron distinguishes between axon regeneration versus dendrite regeneration. If you cut a dendrite, it doesn't regrow the axon. If you cut the axon, it doesn't regrow the dendrite. And this is, I think, really interesting because so far what we know about injury detection is that calcium is involved in the detection of both axon injury and dendrite injury, but at some point the cell has to be able to distinguish between those two things. And we haven't quite figured that out yet. But Sydney's work really highlights the fact that at some point, the cell does know whether it's been injured to an axon or a dendrite. If you cut the axon really close to the cell body, the neurons will convert one of the dendrites into a more axon-like structure. So the polarity of the cytoskeleton switches, and one of the dendrites starts growing really long and kind of looking slightly more axonal in character, but it doesn't seem to go the other way. So if you cut the dendrites, that doesn't seem to prime them for axon regeneration, as far as I know yet.
Hi. Hello. Hi. So I was wondering, you showed that after a single injury, there would be shrinking of the cell because it couldn't grow to a certain extent. I was wondering, with successive injuries, would it grow to the same extent always, or would it progressively shrink over time?
You know, that's a really good question. It kind of gets at the idea of is there like an upper limit to regrowth, or do successive injuries kind of each occur independently? So far we haven't done those experiments in a way that is really kind of reproducible, that I would be able to give you a clear answer. When we're looking at the regrowth after injury in a developing system, it's a bit complicated because the way that the neurons respond to injury over time changes. The younger neurons regenerate a lot more robustly. The older neurons regenerate a lot less robustly. And so it's kind of at baseline, they have a different response. What you would want to do would be to look at a system where the regeneration was the same kind of at two different time points, and we haven't quite figured that out yet. But I really like that question. It gets to the idea of kind of how much do these neurons remember about the fact that they've been injured. What I can say is that, so these neurons, if you injure them during larval development, they'll regrow a lot, and that's a lot of what we showed. And then Sydney alluded to the fact that then these animals go through metamorphosis, and during metamorphosis, they lose all of their dendrites and they regrow a whole new dendrite arbor for the adult, in part because the structure of what this dendrite arbor had to do during the larva was very different from the adult, right? The structure of what a neuron needs to receive input from is different at different stages. And so when we look at neurons in the adult and we compare ones that were injured during the larval phases versus ones that were uninjured during the larval phases, in the adult, they look exactly the same. So that suggests to me, at least, that these neurons have the capability to fully regrow an arbor that looks totally normal, whether or not they were injured first. It depends on the kind of processes that happen later. But yeah.
So you showed like a single cut on a neuron would help the regeneration. Are you looking at ways like, because multiple neurons could be injured in a certain part? Like, are you looking at ways like, besides just cutting individual dendrites, or like a multiple target kind of system?
There are tools out there to do that. So one of the undergrads who's one of my mentees, for a while, she was working on a way to do like whole animal injury to the neurons by causing them to have oxygen deprivation for 15 minutes. And that causes them to shrink their dendrite arbor. And then they also seem to mount a similar regenerative response where they grow back and have more than an uninjured neuron. It's a very small data set, so unknown if it's reproducible, but that is one way that we've been able to do sort of like a whole system injury. Similarly, there's a new tool that came out that we're playing around with. I'm also mentoring some undergrads to play around with, where you can induce expression of this light activated caspase. So that's like a cell death protein. And you can basically turn on a blue light. It turns it on. It causes the neurons to be injured and their dendrites to be injured. And then however long you turn it on for is how much damage you get. So you can kind of control it. It's a bit leaky. So we're playing around with it to see if we can do that kind of on a whole, like on a bunch of neurons at once. So that is something that we're looking at. Yeah, but not so far, still preliminary.
Great talk. And thank you for being here. Really quick, just curious as to why flies were chosen as a living organism to test this, and the next living organism, and why.
So, in part for technical reasons, because we can really beautifully watch all of these dendrites as they are growing and developing, and we can compare how dendrite development compares to regeneration. right? When we started off this project, we knew next to nothing about dendrite regeneration. And so we wanted to start off with a system where we knew a lot about dendrite development. So I didn't show you the PubMed snapshots of dendrite development, but there is quite a lot known in terms of branching patterns and extension factors and transcriptional regulation and all of those kinds of things in these particular neurons. So we started off with this system because we knew not much about dendrite regeneration, but a whole lot about dendrite development. And so we could figure out where were some similarities or differences between how neurons initially develop and then how neurons regrow. I will say that the things that we've found in this system do seem to be true in other systems as well. So since we started off in this system, people have more recently looked at dendrite regeneration in other organisms and what seems to be true across the board. It's been looked at in the central nervous system in zebrafish, for example, and in the peripheral nervous system of other species, not yet in mice, but in C. elegans and other species. And what seems to be true across all of them is that dendrite regeneration is robust, but messy. So across, even in the spinal cord of zebrafish, if you damage dendrites, neurons can initiate regeneration and regrowth, but that regrowth is disorganized in various different ways. So I think the reason why we started off here in Drosophila is because we had the ability and the tools to ask these basic questions. Long term, at the end of the day, we want to see what we're finding in Drosophila, how it compares to other systems. So we could go to neurons in a dish, but I think neurons in vitro are somewhat artificial. A couple of other systems that we're exploring could be looking in organoid systems, so in more complex cellular tissues. How can you ask questions about dendrite regeneration? We could look in organotypic slices, so if you take slices of the brain and you culture those, can we damage those dendrites and look at regeneration? Or in theory, in whole animal models through collaborations with people who are looking at neuron structure and function. You can imagine in the cortex of a mouse, if we're looking through a thin skull preparation, can we damage those dendrites and ask questions about dendrite regeneration? So Drosophila is not the end point, but it was definitely the starting point where we had the ability to ask very precise questions about the cell biology and get some preliminary understanding about kind of fundamental principles.
So I would maybe jump in there and just add one thing, because I think that was a great explanation. I always feel kind of defensive of model systems because I think they don't always get the credit that they're due. And without politicizing this, one could imagine a certain election campaign several years ago where this became an issue, right? And studying in model systems was really kind of mocked as an aspect of science. And that's a problem because you don't know what you don't know, right? And the only way to ask the right questions sometimes are to go back to the most basic system you can imagine, where you can really tease apart what's going on in a concrete way. And so in that context, because we're in the Stem Cell Research Center, I just want to turn and connect two things together, right? So there's that, my defense of model systems as a scientist. And two, I think there's something really interesting in the polyglutamine repeat idea and the concept that what you have here is an animal that is going to go through this complete change in multiple sequences as a part of development, right? Why do I think that's cool? So Ali Bravanlu, who we've had here as a guest speaker in the past, a couple of other people in the field, make the argument based on stem cell research that neurodegenerative diseases, at least some genetic ones, including Huntington's disease, are not a disease of the adult at all. That they are absolutely a disease that starts in development, and we only see it in adulthood. We can't ask that question in a human. We can't necessarily ask that question particularly well in a mouse, or at least no one had, right? And the way that he's asked that question is using stem cell systems, using embryo systems, right? And just like what you showed, where you have this profound difference in the dendritic arbor that shows up in these very early neurons that has to do with polyglutamine repeat, you can see profound differences in the stem cell population, right? During very early development that has to do with those genetic neurodegenerative diseases, Huntington's disease specifically being one that links them together. And that's pretty freaking cool. The idea that we can see that, and emphasizing that you need to go to those systems sometimes to be able to ask that right question. That's my editorializing.
Yes.
Now.
So a lot of folks with diabetes have peripheral neuropathy. Can you model that in flies and give them too much glucose? Do they have dendritic problems?
We haven't done those experiments. I think that some of the experiments that Sydney alluded to that she was working with her undergrad, where we were looking at low oxygen or hypoxia-based conditions, would be maybe the closest that we've gotten so far. You can look at, you can feed flies different foods where they might have lower or higher glucose levels. I think that some of the systemic approaches are hard to effectively model in Drosophila. I think the kinds of diseases that Sydney mentioned, where there's a single gene that is expanded that you can express in the cell types, are a little bit more straightforward to model in Drosophila. I think that I would want to just be very cautious about making direct correlations between a fly model of diabetes and a person who experienced diabetes.
And for those in the audience who didn't open their laptop, I did a PubMed search on dendrite regeneration, and now there's 25 publications, 3 of which are Katie Thompson peers, which means that more than 10% of the literature is you're on your way to success. So
indeed, yeah.
Great work.
Thank you.
Any other questions? Oh, one more.
Sorry, this is more of a broader question and not necessarily related to this, but I was wondering that just like on your path to becoming a researcher, what were the sort of traits that you thought helped you the most along the way, and what sort of mindsets were important along the path?
This is the advantage of the Peter Thompson introduction style.
Do you want to talk about traits, and then I can talk about traits?
Yeah. I feel like perseverance is really important. My undergrad, getting involved as soon as you can in any sort of science is really helpful, because just seeing the process of how science is actually done is really helpful, because there's a lot of failure. There's a lot of things that go wrong. So being able to kind of push through that and like try something new and be willing to try again is like a really strong trait. In my undergrad, I did a project that involved like thousands of PCRs, and sometimes things didn't work, and then you have to just try and try again. So I think that's a really important trait. And I think also being able to kind of prioritize things. So if things aren't working, or if something is like bothering you, just, put it away and move on to something else that's exciting. And being able to like recognize when you like, that's too much, I don't want to worry about that anymore, so I'm going to do something else, and then I'll come back to that. And then you'll be able to kind of look at it with a fresh perspective. So I think perseverance and knowing when to stop doing something for a little bit and move on to something else, so that you can go back and like look at it with new eyes.
Okay. I mean, I agree with what Sydney just said. I think that those are very important characteristics. One thing that has been important for me and my scientific journey is not being afraid to ask scientific questions that nobody else is asking, right? So having that kind of curiosity about something that other people aren't talking about. Science definitely has an inertia and a momentum to it, where there's some questions that there are a lot of people researching, and that they can definitely be very interesting and impactful questions. But that doesn't mean that those are the only questions that are around there. So, and I'm going back to this whole idea of there being 3 publications on PubMed about this topic, and just having the confidence to really investigate something deeply that not a lot of people are looking at, just because the technical tools really hadn't been around in order to ask questions about this part of basic biology. So I think as Sydney put, the grit and perseverance and knowing when to put something aside, but then also kind of having that curiosity to see like, what is the unanswered question? Like being able to, maybe another way to put it is being able to identify the knowledge gap that you are interested in and devise doable experiments that will lead you one step at a time closer to an understanding. Because I think it's very easy to kind of come up with big picture ideas but it's a different skill to be able to kind of break that down into small pieces and figure out, okay, what can we do in the next 6 months to get closer to understanding the cell biology here? Or what can we do in the next year? How can we figure out what is a tractable question and design experiments to help you chip away at that kind of lack of understanding? So, yeah.
Yeah, that was a great summary, right? So if you would listen to Ramoni Cajal, from 100 years ago, who could only look at slices in a static system, right? Mm-hmm. For 100 years, nobody asked the question that you asked because he said no.
He said
right?
No. Yeah.
So with that, can everybody thank our speakers tonight? Thank you guys so much for a really wonderful set of presentations. And have a good night.